Good morning, everyone. Hope everyone is doing very well and enjoying the symposium today. We have been spoiled by Henny and his team to have some lovely speakers in a lovely venue, and we are going to have some engaging discussions. So, firstly, I would like to acknowledge the CEO of Biosafety South Africa, Dr. Henny Kronenfeld, the Kenyan regulatory representatives, as well as representatives from DSI, experts, and stakeholders within the field. A very good morning to one and all. My name is Priyen, and I am a senior researcher at the CSR, and I'm going to be your chair for this session entitled Genome Editing in Practice. And we're going to keep the momentum as Lindy has laid the foundation for us. So thank you for that, Lindy. But our first business of the day is indeed a public service announcement, seeing that I work for government. It is my privilege and honor to wish Dr. Justin happy 40th birthday. So here's Dr. Justin, if you can please stand up. There you go. Happy birthday, Justin. All the very best to you. So today I'm going to start off our session with a true story, and I want you, and I implore you to please humor me for just a moment. A few years ago, I kept seeing colleagues and friends walking around with these earpods with no cords connected to their phone, and it just bewildered me. You know, how is this possible? You know, I'm the kind of guy who loves earphones and I love it to be connected, you know, and that's how I move. Anyway, time went on, and as you can imagine, you know, us scientists, we sometimes are, what's the word, recalcitrant towards technology upgrades. And so eventually I had to upgrade my phone, and so I did. And I was very happy. I was going to go home and listen to some music, and I opened my box of my new phone, and I looked. I kept looking for the earphone jack, and I couldn't find it. It was at that point, lo and behold, that I soon realized that with matters of science and technology, change seems to be the only constant. Our first speaker today, ladies and gentlemen, is Dr. Manuela Campa. Now, she is a researcher in the genetics department at Stellenbosch University and has an extensive background in plant molecular biology and technology. She received her education in Italy and gained experience in various international labs before joining the Wits lab in 2014. Now, her current focus is on the use of genome editing to enhance grapevine tolerance to biotic and abiotic stresses. Ladies and gentlemen, please kindly welcome our first speaker for today's session, Dr. Manuela Campa.
First of all, I would like to join Henny in expressing my enthusiasm of being here today, sharing this day with all the genome editing community, sharing our frustrations and our excitement. So, what I'm going to do now, and I want to ask, sorry for the expert in the field, in genome editing technologies, because I'm going to be very basic and simplistic maybe sometimes, but I think that's what was requested by a survey, to give a little bit of a breakdown of an introduction of how this technology works. So first of all, genome editing allows us to make precise modification of the genome of an organism or a cell. And genome editors that can do that have been around for quite a few years now. And they are meganucleases, zinc fingers, and talents. And they have done a very good job, they've been successful. But the real change and the reason why we're all here now, we're here today talking about genome editing, is CRISPR-Cas. That really revolutionized the whole field. And here you can say the increase of number of publication with CRISPR technologies compared to the other three previous or first generation gene editors. Not that they are not good, they're nice and efficient, and we will hear from one of the presenters later how one of these, the talent, can be successful. But anyway, CRISPR is the revolution, basically. And how does it work? First of all, has been discovered in bacteria as their natural immune system. And, but the real change happened in 2012 when, as Roy said earlier, Jennifer Doudna and Emmanuel Charpentier harnessed and made of these bacterial immune system a tool for genome editing. So that was the real big change and step that allow now all of us to being able to edit so easily. And that is one of the main characteristic and points of CRISPR, is that it's simple. Okay, why is it simple? So there are two components, the protein, the Cas9, the red blob here in the slide, and the RNA molecule that we call guide RNA. So the guide RNA is what drives the Cas9, the Cas, to the DNA, to the specific target that we want to edit, okay? And once the guide has found the specific target, then the Cas9, or nuclease, cuts the double-strand DNA. At this point, what happens is that the natural cell repair system of the cell take over. And most frequently, what happens is the homologous end joining repair mechanism occurs for most of the cases. And this causes indel mutation, which means insertion or deletion of one or two bases or small bases. And this causes knockout, so they turn off a gene most of the time. And I will say that these correspond to the SDN1 of the regulation we were talking earlier. On the other side, much less frequent, is the homology direct repair, which require a donor template and the homology of this donor template with the cutting site. And then in this case, we have an insertion, so a knock-in. And I think here we are in the case of SDN2 and SDN3 that we were talking later. Okay, but how do we go about performing a CRISPR project? So the main, I would say, phase is the first crucial step of designing the experiment. And what is it necessary to design the CRISPR experiment? To know about your target, your gene that you want to target. So nowadays, I mean, most of plants and organisms have been sequenced, and it's very easy to have access to the genome or the sequence of the gene that we are interested in. But it's also important to resequence, to be sure that what you're targeting is what you know or you think it is. And why is that? Because being CRISPR so specific, if by any chance with the cultivar, the variety that you're working with, has one nucleotide change, CRISPR won't work. But anyway, this is one practical aspect. And then you need to decide, once you decide what you want to target and you know all about it, you need to decide, do I want to target one gene? Do I want to target multiple genes? Do I want to target that gene in multiple positions? And this is all possible with CRISPR. It's another important characteristic, the multiplexing capacity. Okay, the next step is to design the guide. And this is made very easy by a few different software that are available online. And here you can see how many every year are developed. So for plants, I'm most familiar and I use quite often CRISPR-P, RGen, CRISPR, and ChopChop. They all are a bit different because they work using different algorithms and they use different data sets for their algorithms. And unfortunately, very few of these are plant-specific. But anyway, what all of them take into account is the PAM sequence, which I haven't told before, but is one of the requirements of the Cas. It's the protospacer adhesion motif, which is absolutely necessary for the Cas protein to bind to the DNA. And different Cas have different PAM. So the most known Cas is Cas9, the first discovered, the most used, isolated from Streptococcus pyogenes, and it has a very specific PAM that is NGG, so N meaning any nucleotide and then GG. But every day new Cas have been identified and discovered. Anyway, there's a lot to say about that. But anyway, so the PAM can be limiting, but not anymore. Then secondary structure, GC content, and off-targets. So off-targets, it's a very important aspect because you don't want your guide to target any other places except your gene. So this software will give you a list of potential of target, considering 0 mismatches, 1 mismatch, 2 mismatches, 3 mismatches. So once you have all this information, you can choose your guide, which is all a very empirical exercise. But there are ways, and I think in the presentation that will follow, we will see how guides can be validated before going through the full transformation process in case of plants. Okay, and then the performing of the experiment. Biggest bottleneck is still the delivery. So the transformation part, the delivery of the Cas component can be still challenging, but that I feel is, I don't know if it's linked only to genome editing or more to genetic engineering in general. So what we need to decide is how do we want to what we want to do, how do we want to deliver these Cas components. We can choose between ribonucleoprotein complexes, RNPs, which means the Cas protein and the guide RNA. So we basically can just purchase these from Thermo and Sigma, order the two components, assemble them together, and just deliver into the cell using PEG or microinjection or electroporation, depending on the explant we are talking with. And this is very important because this is one of the ways to obtain DNA-free, so not, in theory, not GMO plants. The other way, more classical, I would say, is a vector. And for these, there is a very useful and amazing resource that is AdGene, is a reservoir, a collection of all the possible plasmids that you can imagine. And there is a special section for CRISPR where all the, basically it works that when a lab develops a vector for CRISPR or for any other application, they can deposit in AdGene. And then every scientist in the world can request, and you just pay the shipping fees. So it's very convenient, and it gives a lot of information, also the cloning strategy and all these. And then third is inserting the Cas component as in vitro transcribed capped mRNA, which is less used in my knowledge. And they can also be ordered and synthesized by companies. This is more specific for plants, and I would like to draw your attention to the protoplast transfection, because it's the one that is most used for DNA-free approach using RNPs. But maybe we can discuss that later. Then we need to evaluate our editing, so the editing analysis, and there are three main ways. One is very simple and quick and cheap. It's using enzymatic cleavage assay that give us an answer as like yes or no, you have the editing or not. So it's a quick screening, but it doesn't give much information. More informative is PCR followed by Sanger sequencing, and there are also these two softwares that can analyze the Sanger sequencing and give an estimate of the type of indels or modification that you have in your cells. And then the most precise is the obviously next generation sequencing that will give us the exact type of mutation and the quantification of the different type of mutation that you have. And here it's them summarized in a table. Obviously, next generation sequencing is the most expensive, although sequencing technologies are becoming cheaper and cheaper every day. And for regulatory purposes is obviously the method of choice. Lastly, phenotype and functional characterization. And here is, I guess, the same procedure as to characterize transgenic plants. Analyzing the eventuality of pleiotropic effect and evaluating if your modification, your edit, actually brought the effects that you wanted. So, as I said, this was a very simplistic and quick summary of what CRISPR is. It's evolving every day. The toolbox is becoming quite expansive, and so there are different. So we talk about the double cut of DNA. We can do a single cut. We can use CRISPR to visualize DNA. We can use CRISPR for epigenetic, and base editing and prime editing are probably the future, because they allow us to create changes that are not only in a precise location, but also the exact edit and changing that we want. And this is it. This is just a summary of, I think these 4 crops are the 4 genome editing crops already on the market, which I think it's quite remarkable for such a young technology to have already some plants on the market. And linking to what Henny was saying earlier, if you look there, almost all of them are quality traits. So the value is in the quality. It's recognized more in the quality, but I mean, this is maybe a discussion that we can have later. Thank you.
Next up on the itinerary is we have representation from the University of Witwatersrand in the form of Tiffany Smith, as well as Deanne du Plessis from the University of Pretoria. And a quick introduction on Tiffany. Now, Tiffany is currently a PhD candidate who is a member of the Antiviral Gene Therapy Research Unit at the University of Witwatersrand, Johannesburg, South Africa. Now, after joining this unit in 2016, she worked on designing obligate heterodimeric transcription activator-like effector nucleases, TALENs, for the inhibition of hepatitis B viral replication. This work generated promising results and was indeed submitted as a master's dissertation in 2018. Her research now focuses on generating talent-encoding mRNA to specifically target and inactivate the hepatitis B virus. Tiffany looks forward to furthering her research and contributing towards the field of medical science using these amazing technologies. Ladies and gentlemen, please welcome Ms. Tiffany Smith.
Good morning, everyone, and thank you so much for that wonderful introduction. It is very much a pleasure for me to be here this morning. As you can see, my talk is going to be a little bit different from everyone else in this session. I will be talking on talents, as Dr. Campa has highlighted, and it is indeed very much, I think, maybe a little bit of a controversial topic. I don't think it falls quite within the GMO specificities, but it's just to highlight the diversity of these gene editing tools. And I think particularly in the developing world, such as South Africa, I know that we are unfortunately plagued with a lot of pathogenic infections. So I think it is important to highlight what work is being done out there in the field. So without further ado, my title is Engineering Talents to Permanently Disable HPV Replication. So just to give you a little bit of a background on the HPV epidemiology, hepatitis B is a potentially life-threatening viral infection that does occur in the liver, and it is caused by the hepatitis B virus, hence HPV. And recent stats, according to the World Health Organization, have actually shown that close to 300 million people worldwide are living with chronic HPV infection. And the greatest burden of infection does occur within the African and Western Pacific regions. This, of course, includes South Africa. So in these countries of the highest, where we have highest epidemic breakouts, the transmission of HPV infection is through contact with blood or other bodily fluids of an infected person. And this is mainly via the vertical transmission from mother to child during birth, either through sexual contact, as well as the sharing of equipment between intravenous drug users. Now, chronic sufferers are at risk of developing liver scarring as well as liver cancer, and this attributes to approximately 600,000 deaths annually. So just to look into why is this virus such a nasty one, I'm not going to go into the complexities of the genome. I just want to highlight key factors as to why we are struggling with these infections at the moment. So the HPV genome is a partially double-stranded relaxed circular DNA, or rcDNA. And upon infection, this rcDNA enters the nucleus of your liver cells. It then harnesses your liver cells' sort of processing to form what is called covalently closed circular DNA, or cccDNA. And this is essentially the little bugger that is very much problematic and very difficult to target. As you can also see, it's very important as it results in the cccDNA is the template for producing the viral transcripts. And yeah, unfortunately, the therapeutics that are available has limited targeting against the cccDNA, therefore leaving it intact, and it is then able to re-establish infection, and that's the current problem that we're facing. It is also very unfortunate that an HPV vaccine has been implemented well over 20 years ago, but infections are still on the rise. And with the current technology that's not able to target the cccDNA, this brings us to looking into novel therapeutics as a way to permanently disable cccDNA. And in our lab in particular, we focus on gene therapy as a mechanism. So like I've mentioned, my focus is going to be on transcription activator like effector nucleases, which is a mouthful. So it's just TALENT for short, and I'm just very briefly going to go into how TALENTs work. So similarly to CRISPR, TALENTs was also found in bacteria. It was actually, this bacteria was found to infect crop plants, and upon further investigation, what was found there was that the effector proteins were found to bind to DNA and was involved in transcription processes. So for the purposes of gene editing, what you do have is you have this tail sequence that is composed of about 18 tandem repeats, and this is the repeats that is necessary for binding to DNA through what is called RVDs, or repeat variable diresidues. So each repeat technically consists of this 34 amino acid sequence that is repeated, and as you can see, the RVD highlighted in this example is HD. So HD would then bind to C in your DNA. So of course, in this particular example, wherever you see sort of the purple oval, that's where you'll have your HD that would recognize your C base. So for the Afrikaans people in the audience, I am going to refrain from pronouncing the endonuclease. I don't want to sound too profane, so I'm going to call it the FOK1, and that is actually what it is called, so that's not a typo. So this is the endonuclease, and it actually functions as a molecular scissor. So this would induce cleavage of your target DNA. So the way that this FOK1 works is that it needs to work in a pair. So what you would then need is you would need two tails that would come and bind to your DNA, and this would induce your site-directed cleavage, which then would result in a break in your DNA. And as Dr. Campa has already highlighted, what we rely on in this case is non-homologous end joining. So you'll come and you'll get a repair, but the repair would be a mutated sequence. And this is ideally what we want in our case, because we want to mutate the CCC DNA. We don't want it to continue to replicate. So this is what we would refer to as a first generation talon. There has been more modifications that has been done in the endonuclease to just offer better efficiency for binding and targeting, and we refer to that as second or third generation talons. So third generation talons are ideally what should work best in a practical setting. Okay, so there's just two technical points that I want to bring out when you are designing a project that requires talons. That, of course, is you need to check where you are targeting and make sure that once you do have that, you then look at, as Dr. Campa has also highlighted, you look at prediction tools, because off-target effects are a big no-no, particularly in gene editing for therapeutic effects. And then these are just some of the tools that I've highlighted on the screen. And so in our lab, we have designed talons that targets the surface and core genes of the HPV CCC DNA. So as you can see, there has been several publications already that shows how effective these talons are against HPV. I am just, for the sake of time, going to highlight some of the work that I had obtained in my study. So these experiments were actually performed in mice, and this was an infection model of mice, and what I first looked at was the hepatitis B surface antigen levels. So this is a marker of active replication, and in fact, when you do go for diagnostic testing, this is what they will assess to see if you do have active replication, and this will therefore mean that you have been infected with HPV. So what you can see from this data is I used the first, second, and third generation talons to see how it would cope in the mice models. And ideally for this set of data, we would want the surface talons to function the best, because it would be targeting the surface gene. But what I did see is that even the core talons still reduced the surface antigens significantly. Interestingly, like I had mentioned, the third generation talons, which should have functioned the best, seemed not to have the best output in this particular result. We then wanted to see whether the talons had any effect on HBV virion secretion. So this is again just looking at what is happening to the virus. Is the virus getting into the liver cells? Is it being replicated? Is it escaping from that? And is the replication an ongoing process? And what we do is we measure the viral particle equivalents, and what you can nicely see compared to the mock, so sorry, I should have started with that. The mock would just be an infected mouse where it is not treated with anything. And what you can see is that with all the talons that were used, you get a significant reduction in the viral particle equivalents. Finally, what we also looked at was pregenomic RNA. Now, pregenomic RNA is very essential in HBV expression. It does serve as a template for reverse transcription, which leads to the relaxed circular DNA. And if you'll remember from the beginning of my presentation, I showed that it is the relaxed circular DNA that gets into your nucleus, and that serves as your precursor for your cccDNA. So if you do have high levels of pregenomic RNA, it shows that you have viral persistence and active gene expression. And again, what we could nicely see was that these talons significantly reduces the pregenomic RNA. So while this did fare well and it produced very exciting results, we needed to look at how can we make these talons more applicable for gene therapy, considering the fact that clinical translation would require large manufacturing as well as dosage regulation. So how do we move on from just testing this out in the lab to getting it out there for gene therapy? So at the moment, these talons have been expressed as DNA, and we do know that using naked DNA or nucleic acid sequences, it does have its drawbacks, which is highlighted on the screen. And so what we've moved on from doing, from trying to get away from using naked DNA is in vitro transcribing our DNA templates to RNA. So how it works is in a cell, you move from DNA to RNA to protein. So also by using RNA, it just skips the step of having your DNA having to enter into the nucleus and then being transcribed. You already have your transcribed product. So one of the advantages you have from moving from DNA to RNA is, of course, there's no risk of genome integration upon entering the cell, because DNA actually can integrate within your genome. And again, very importantly, with your RNA, it is easily scalable as transcription can take place in vitro. So this is just sort of in a test tube. And then very importantly, the delivery aspect that Dr. Campa has highlighted on. Once you have RNA, you can then very easily deliver this with lipid components that is biodegradable, and you can, in fact, do this on a large-scale synthesis. And I think it's also important for me to note that this is essentially what the COVID vaccine is composed of. It's composed of an RNA sequence that is encapsulated by this lipid, and it has proven to be very efficient. Okay, so I have actually been able to successfully convert the DNA to RNA, and this is just showing the expression of the talons. Again, in the mock, that is just cells that have not been treated with the talons, and this is in cell culture this time, this is not in mice. So all the green cells that you see there is just a tag that I've inserted to be able to detect expression. And again, very nicely, I am able to see a significant reduction in the particular marker that shows whether you have infection or not. So that sort of brings me to the end of my discussion with my results, and what I just want to highlight is there are other uses for these talons. You don't particularly have to use them as nucleic acids or have them in sort of pairs. You can use a single tail fragment, and instead of having the FOK1 attached to the end, you can use an epigenetic modifier, which would, instead of cutting, it would repress your expression. And you can also still go ahead with using the talons as DNA, and instead of packaging it with the lipid components, you can package them with viral delivery, which an example is AAV. This has also been used in the COVID vaccines. Trouble areas, I think everyone will probably highlight on this, is certainly the off-target effects, and very much so in my setting in gene therapy. You want your talents to be as specific as possible. It should only target the HPV genome and nowhere else in your human genome. There are some design shortcomings because what you see in theory might not always result to what you would want to see in practice. And that is particularly what we saw with the third generation talents. We thought that this would, in theory, has been the most adapted and should be the most efficient. Turned out not to be. In fact, it was the second generation that turned out to be the best. So we are still looking at trial and error, and we are doing this in animal models and in cell culture models before it even gets to any sort of clinical trial. And yeah, with that, I'm going to come to an end and just acknowledge the following people that made all my work successful. Thank you.
Thank you so much, Tiffany, and we look forward to the non-human primate clinical trials hopefully coming up the ranks. And thanks for the representation on the prospects of these genome editing technologies in precision medicine. So we have Deanne here, who's going to be presenting on more genome editing work in microorganisms. So Deanne, I would like to kindly have you to please introduce yourself to the audience, and I'm going to load up your presentation.
Yes,
is that fine?
Yes,
okay, cool.
Hello everyone, it is a privilege to be here. I'm a master's student at FABI, University of Pretoria, and I'm very excited to talk today about CRISPR in fungi, especially how we use CRISPR to study the function of genes. So my project is about characterizing a gene we think is involved in the virulence of a pathogen. And the pathogen which I'm working on is called Leptographium wagneri, and it is the causal agent of black stain root disease. Now, this pathogen is able to infect a single tree, and it is then able to spread to the neighboring trees through root-to-root contact. And this will then lead to a circle of infected trees, which will over time start to wilt and die. So most species within the genus Leptographium is considered to be non-pathogenic or saprophobic. So we wanted to know what makes Leptographium wagneri a pathogen. To do this, we sequenced and compared the genomes of Leptographium wagneri to its non-pathogenic relatives, and amongst the genes which are only present in Leptographium wagneri, we identified a putative lacase gene. Now, fungal lacases have been well studied and they are involved in many processes. However, their most prominent role is in the decomposition of lignin. We also know that this lacase is upregulated within Leptographium wagneri. So we then wanted to know, could this lacase potentially contribute to its ability to proliferate within the xylem, seeing that it is a vascular wilt pathogen. And to do this, we generated lacase knockout mutants using CRISPR-Cas9. So there are three main stepping stones when starting a CRISPR project in a fungi, the first being the design and synthesis of your single guide RNA. Now, this will essentially be the guide that takes the Cas9 to your target gene. So the most important is to make sure that there's no off-target effects. You don't want it to be taking your Cas9 into any other areas in your genome, as well as the structure. So here we can see the typical centroid structure fold, which is your single guide RNA and the scaffold. And it's just important that this structure is sort of like this because it enables the construct to move to the donor, to the gene of interest and actually be able to bind. And then finally, you also want to make sure that this is actually able to cleave in vitro. So here we can see the amplified lacase gene as well as a digested product, because you don't want to go through all the trouble of transforming and then only realizing at the end that your single guide RNA wasn't actually able to cleave. So after the Cas9 has created a cut in your organism's DNA, this can then be repaired through homology-directed repair if you have a donor DNA construct. So the positive thing about having a donor DNA construct is it can enable marker-assisted selection. So this is where you could design your donor DNA, that it has flanking regions to your gene of interest, and then this will then facilitate homology-directed repair. This is actually quite easy to synthesize in the lab. You just amplify the flanking regions of your target gene as well as an antibiotic resistance gene, and then you fuse this together in a simple fusion PCR. It's also important just to confirm that your donor DNA construct is what you think it is, and we do this by amplifying and sequencing various regions of the donor DNA construct. Then, as previously mentioned, the delivery is quite a big step in transforming. So because fungal cells also have a cell wall similar to plants, what we do is we generate protoplasts. So these are cells without a cell wall, which are more like red, they are more accepting of taking up foreign material. So what we do in fungi is we collect the spores. Here we can see that the spores of Leptographium are these teardrop structures, and then we incubate that so that it forms a germling solution, which we then use in a lysing enzyme mix to form the protoplasts. And here we can actually see that the protoplasts are these round shape after they have lost their cell wall. So then we can move on to the actual transformation reaction. Now, when working with fungi, most of us work with non-model organisms, so the simplest way to transform is to assemble an RNP complex or a ribonucleoprotein complex, and this is basically just combining the Cas9 protein with your single guide RNA and then using that in your transformation reaction. Then the physical transformation reaction is where you combine the RNP complex with your donor DNA as well as your protoplasts, and then we let that solution regenerate overnight. Then we finally can get to the selection of your potential transformants. So what we do is we plate out our transformants on a nutrient-rich media, which we call OCMA, as well as a nutrient-rich media containing the selective antibiotic marker. And here, if we look at the results, we can see that the protoplasts which were plated out on the normal media showed a lot of colonies forming. The media actually started to turn brown, whereas the protoplasts which were plated out onto the selective media showed less colonies, which we can see there on the plate. Then the screening process actually also involves that you have to confirm that your target gene has been knocked out. This can be achieved through PCRing as well as sequencing. And if we take a look at the sequencing results here, we can see that there were no reads aligning to the lacase gene, confirming that this gene has been successfully knocked out. However, what we did see was there were quite a lot of reads aligning to the donor DNA construct, indicating that this construct has integrated into the genome multiple times based on the high coverage. And then if we just fast forward to some of the pathogenicity trial results where we inoculated Pinus patula seedlings with Leptographium wagneri, we can see here that the trees that were inoculated with Leptographium wagneri showed the typical symptoms of black stain root disease, which is stunted growth as well as yellowing of the leaves and wilting, and these trees did eventually die. Whereas if we look at the trees which were inoculated with the lacase knockout mutants, they appear to be quite healthy compared to the mock inoculated control. So this then leads us to believe that this lacase gene actually plays quite an important role either during the initial infection stages or perhaps during the proliferation of this pathogen within the xylem. So just some basic things to consider, which I think is most important, is the the design and synthesis of your single guide RNA. You want to make sure that it is specific, as well that it has the correct structure, and its cleavage ability is quite important. And then, with generating protoplasts, if you're working in a non-model organism, this process has to be optimized quite a lot. As different fungi have different cell wall components, so you might need to try different digesting enzymes, different digestion times, so this can be quite a tricky process. And then also the selection process has to be optimized for the species that you are working on, as each species has different levels of sensitivity to the antibiotic that you will be using. But other than that, besides a lot of optimization, it's quite fun to work with CRISPR in fungi. And with that, I'd just like to thank everyone who contributed to this project and to make this possible. Thank you.
Thank you so much, Deanne, and I know that Dr. Tuan Duong is very proud of you, so thank you for presenting and sharing your work here today with the community. Really, really appreciate it. Colleagues, we're moving on to the next theme within our session, and that is the CRISPR-based genome editing in plants. And here we have representation from Luke Kim. Now, Luke is an MSc student in the Forestry Molecular Genetics program at the University of Pretoria. His research is focused on enhancing our comprehension of wood formation in economically significant tree species, specifically eucalyptus. Now, utilizing CRISPR-Cas9, his research is dedicated to functionally characterizing genes essential to the wood formation process. Now, based on this experience, Luke would like to share the practical know-hows of CRISPR experiments being performed in these woody plants. Ladies and gentlemen, please welcome Luke.
Okay, what a stimulating day. We're moving from virus to fungus now to plants. And I'm going to just try to shift gears for a bit. So I would like to share the journey of my MSc research, but I would like to specifically focus on the practical challenges that I encountered and overcame through the journey. And my MSc was, what we wanted to explore through my MSc was to explore the biosynthesis, to understand the genetic mechanisms underlying biosynthesis of wood using CRISPR. So without further ado, to give you a brief introduction to my project is that, so the forestry industry contributes significantly to the bioeconomy of South Africa. And in an attempt to aid this economically significant industry, we wanted to understand the biology behind wood formation and also wood processing. Through the previous research in our research group, we identified a number of candidate genes that are putative, that are thought to be involved in wood formation. And we were interested in xylem biosynthesis. Xylem is one of the most abundant constituents of secondary cell walls of the trees, which then forms wood. So we wanted to understand what is the biological functions of these xylem biosynthesis genes in wood formation. And we also wanted to gain a better understanding of how the genetic manipulation of these xylem biosynthesis genes could affect woody biomass processing. And of course, naturally, we encountered many problems, and I would like to share 3 with you today. Hopefully that we could all benefit from it. So as Dr. Campa mentioned, that the single guide RNAs are a critical aspect of CRISPR experiment. And yeah, so there are a lot of plenty of tools, online tools that are available to us freely. So for example, CRISPR allows not only for you to design single guide RNAs, but it also provides the predictive scores of the efficiency of your guide RNA and potential off-target effects. And because I'm targeting multiple xylem biosynthesis genes, I've designed multiple guide RNAs, which then are multiplexed. And before we jumped into implanter assays, we wanted to test the efficiency of the guide RNAs using in vitro assay. So it's the same as what Deanne just mentioned in her slides. Luckily, some of our guides were really good. They were able to induce full digestion of our target sequence. But for some, they were able to digest the target DNA sequence, but we did see a partial digestion because we still had a hint of our target DNA. So it didn't change whether we increased the reaction time or it occurred multiple times whether you change the experimental conditions. So we assume that there's a variation in the efficiency of certain guide RNAs. And it's really important, and from this experience in the future, we would like to, and we are going to, and take this efficiency, or the validation step critically, because in the later stage, in an implanter experiment, we did see that the efficiency actually affected the implanter mutation in our model species. The challenge number 2, which is the biggest challenge that we had, is the transformation of the plant materials. So what it entailed was we transformed our CRISPR-Cas construct into the agrobacterium, which then were cultivated with the poplar leaf discs, which then were grown on selective media. So only the cells that harbored these CRISPR-Cas construct were allowed to grow, form callus, and grow shoots. This process had very low efficiency, ranging from almost no successful outcome up to 2.4% success rates. And we also learned along the way that you really need to optimize your media depending on what system you're working with. So it doesn't matter whether it's coming from at the university, whether it's coming from at the literature. It's essential that you need to optimize your media. You can change different medias. You can try to change the concentration of the hormones that you're putting in. You can try to change the concentration of antibiotics that you are using for your selection. And also, this is a famous saying in our lab, there's always success in mass. So if you want to do transformations, especially with plant materials, because it has such a low efficiency, we would definitely go with more than cutting 200 to 300 leaf discs at a time to maximize your efficiency. And yes, so we had putative plants that we thought are to be mutated through our CRISPR system, but we can't say that with confidence without actually confirming their actual DNA sequences. So first, we took amplicons and we processed them, we sequenced them using Sanger sequencing. Sanger sequencing was quite limiting in our case because it could only indicate whether there was a mutation or no mutation. But we wanted to get a much comprehensive insight into the spectrum of our mutation. So, for example, the allele specificity of the mutation. Also, we wanted to gain we wanted to see the different types of mutations. So we employed another sequencing mechanism, which was Illumina MiSeq, as again highlighted by Dr. Campa. And it was actually really great. I know she said it cost a lot, but I think it's a really worthy investment because through Illumina MiSeq, we could tell the allele specificity of our mutation, and because the MiSeq gives you a number of reads, we could understand the extent of the mutation. But the most striking observation that we had was that we found genetic